Hello, I'm Mahica, one of the bio teachers here at ACE. Today I'm going to be going through recombinant DNA and the main aspects of human insulin production from E. coli bacteria. This is going to be a really important part of Module 6 and one of those biotech that is quite commonly tested in the HSC. So going through recombinant bacteria, it's really important that we understand the process of how insulin is going to be made using E. coli bacteria. This is a common transgene and a transgenic organism used in the medical industry. Firstly, we have to be able to identify and isolate the insulin gene of interest. Fluorescent in situ hybridization is going to be a great technology that allows us to visualize the insulin gene inside the human gene. From here, we can use specific restriction enzymes to cut out this gene, which can make sticky ends and an isolated gene product. Furthermore, these same restriction enzymes will be used to cut the plasmid such that this recombinant gene can fit inside our plasmid and make a recombinant plasmid. After that, we're going to be using DNA ligase to join up the plasmid together. At this point, it's going to be important for us to recognize which of these plasmids have actually undergone this recombinance process and which ones have just closed up back on each other. This is going to be where we use a reporter gene. A reporter gene is going to be a specific gene that sits at the site of insertion of this recombinant transgene. When the reporter gene is going to be broken, we can understand that something else has been put in its place. So we have a transgenic organism. In this case, we can use something like a LAC-C gene. LAC-C genes traditionally give plasmids their blue color. So if we're going to be having an insulin gene inserted at its point, the LAC-C reporter gene should be broken and therefore there shouldn't be a blue color. We can use this to distinguish between our recombinant plasmids and non-recombinant plasmids and keep only the recombinant ones. From here, we want to put these recombinant plasmids into our bacteria. This is going to be a process called transformation. There's many ways in which transformation can occur, the most commonly being heat shock and electroporation. Both of them have similar mechanisms in which they make the cell membrane a bit more transmissible, so plasmids can move easily into bacteria. Recall that plasmids are going to be sections of small circular DNA that replicates independently from bacterial chromosomes. Once these plasmids undergo this form of horizontal transfer into one bacteria and another, they can undergo replication and use their normal transcription and translational processes to make insulin. Insulin will be removed from the bacteria because it's not a normal product of the bacteria, and we can externally harvest it and use it for synthetic uses. Examples of uses of insulin include for type 1 diabetic patients who can't produce insulin on their own. So that was a quick overview of recombinant DNA and how insulin can be made using transgenic E. coli. Make sure you revise your other biotechnologies and keep following along for more content from biology.